Hello and welcome to our program this evening or this afternoon, wherever you may be, or in the case of our speaker, Yeung, good morning. She's beaming in from Hawaii. Thank you so much for joining us. My name's Sam Ankerson. I'm the director of the Bailey Matthews Shell Museum, and very much looking forward to Nori's program tonight here in Southwest Florida, where when we think about mollusks, we're often thinking of marine mollusks. And so we love being able to have this focus on land snails, some of the many mollusks that live on land, and then the particular situation of Hawaii and what's going on there, which to me is just so fascinating, given obviously the isolation of the islands, but as you'll hear from Nori, the incredible diversity and unique issues facing the populations there. So we're honored that she is with us to share her expertise and her experience, and also a look to the future for these animals in Hawaii. For those of you who are tracking the progress of the Shell Museum and our rebuilding, just a quick note of update, which is that we're deep in reconstruction right now. The museum is frankly a construction site, and with a lot going on. So the good news is contractors are hard at work. The ground floor, a lot of work's been done to reinstitute the walls and ceilings and electric, and actually beginning next week the aquariums start to get reinstalled, and that's a process that'll take several weeks, and we're hoping then to begin bringing in live animals in the sort of September-October timeframe. So that's where we are. We're aiming to have part of the museum, the aquariums, open in November or December, somewhere in there, and then the Great Hall of Shells and the second floor exhibits will follow in 2024. So thank you for your ongoing interest in that and your support, and we'll continue to update you as we move along. This talk is the first of our summer series of online lectures, and I'll just let you know quickly about the ones coming up. On Thursday, August 17th, Dr. John Pfeiffer of the Smithsonian National Museum of Natural History will be giving a talk on freshwater mussels. On Thursday, September 14th, I'll be giving a talk on the plans for the renewal of the museum. That date lands quite close to the 1-year anniversary of Hurricane Ian, and it'll be kind of a report on the museum and a look to the future and some of the changes that we're going to be making. And then in October, on October 12th, our own Dr. Jose Leal will give a talk called The Charisma of Cowries. And these are all free, and you can register for them on our website, shellmuseum.org, and they're under the education section. And there also, as always, you can access the archive of recorded lectures, which now numbers over 20. And there's a great range of programs there for you to enjoy free of charge. If you have with questions for tonight's program, this is the webinar Zoom format, so the chat function is the way to ask questions. So that's if you move your cursor along the bottom or wherever the Zoom icons are on your screen, and ask your questions by using the chat function. We will be glad to get to those at the end of Nori's talk. And with that, I will be, it's my pleasure to introduce Dr. Norine Yeung, who has been the Malacology Curator at the Bishop Museum in Hawaii since 2015. She's also a Natural Area Reserves Commissioner for the Department of Land and Natural Resources in Honolulu. She's been involved with Hawaiian land snail conservation research and publication for over a decade, She earned her BS, MS, and PhD in zoology from the University of Hawaii. And she's also important to us, a good friend and good colleague of Jose Leal, and a great contributor to our field. So, Norine, thank you for being here, and the Zoom floor is yours.
Well, thank you for inviting me. I really appreciate it. And I really enjoy visiting you guys, and I hope I can visit you guys soon again. With COVID and all of the repair work, it's been pretty tough couple of years, but hopefully I can make it there back very soon. All right. So yeah, so please put any questions or comments in the chat, and we'll try to look through it. And I'm just going to give you a quick overview of some of the things that we're doing in Hawaii, as well as the conservation status of our Hawaiian Land snails. Okay, so let's just get started. All right. So, right, just making sure. All right, everybody can see this title page. I think so. All right.
Yeah, we can see it. That's good.
Okay, great. All right, thanks. All right, so I just wanted to start this, as we all know, we're in a biodiversity crisis. It's not really new to any of us, so I'm not really going to hammer this topic home. But yes, we're in the age of extinction, with a lot of things going extinct faster than we can even study them. And throughout the world, there are about 36 biodiversity hotspots, and biodiversity hotspots are areas that have high species diversities and endemicity, but also a very high extinction rate. And so in the Pacific, we have about 6 primary hotspots, but the ones that encompass most of the Pacific Islands are these 3: Micronesia, Melanesia, as well as Polynesia. And so these hotspots contain more than 6,000 islands, and it's about 1.6% of the land area of the continental US, but it represents 14 countries, as well as 3 US territories, which includes the Marianas, Guam, American Samoa, and the state of Hawaii. And Hawaii, of course, unfortunately, has 500 species under Endangered Species Act, and there's about 600 total on this list. So we have the title of the endangered species, as well as the extinction capital of the world. So these are things that we don't really want, but unfortunately we do have these titles. And so there are a couple of major threats to biodiversity, and you guys probably know this, and most of them are are actually human induced. We have habitat destruction, we have pollution, we have impacts of invasive species, and then also all the diseases that are now associated with all of these other threats, including climate change. But I usually like to add another threat to biodiversity. And this is what I call the knowledge shortfalls. And we have a lot of knowledge gaps in terms of conservation and understanding the world, to the extent that if we don't fill in these gaps, there's no way that we are able to help solve the problems of overexploitation, climate change, habitat destruction, or invasive species. And here are some of the general topics for these shortfalls. So we have the Linnaean shortfall, which a lot of the museums are trying to fill in this gap. It's the species taxonomy. What species are out there? What are their names? And then we have the Wallacean, where are they distributed? What are their ranges? The Prestonian, how many are there in a population? How many are there in a species, right? The Darwinian, how are they related to each other? Right, and then it goes on to the Austromian, where it's the needing to fill in the educational and knowledge gaps of the community. And so if we do want to conserve anything, whether it is snails, plants, ecosystems, there are a lot of subjects and topics that we need to engage to do, right? So, but however, you can't really understand evolution or ecology or understanding any biotic interactions until you start here first. And that's why museums are so important. We are basically the libraries, right, of life. We help categorize, catalog, and help identify what's out there. Until we know what's out there and how we can tell them apart and the relationships, we can't understand anything about their life history or where they live. Because if you don't know what they are, how can you actually start monitoring all of these other requirements? So what a lot of people don't realize is that out of all of the animal groups, mollusk has the highest extinctions from the 1500s, higher than birds and mammals and reptiles. And most of these extinctions are actually happening in terrestrial ecosystems, with the majority of extinctions happening on Pacific Islands, like Hawaii, right? So Hawaii is about 1/1700 the landmass of US and Canada. And the US continent has about 1,200 to about 1,400 land snail species that we know of. Hawaii has almost 800. And all of the species except for 3 are only found in Hawaii. So they're nowhere else in the world. And those other 3 are endemic to other Pacific Islands. So you'll never find them on any continents either. So we have a very special land snail fauna in Hawaii that if they go extinct, there's nowhere else in the world that we can find these snails to recolonize these islands. So something cool to know about our Hawaiian land snails is that the migrants or the species that came to Hawaii didn't only come from one small group, okay? So this is the Stelomatophorine phylogeny, what we call pretty much the land snail tree of life. Most of the land snail families belong to this group, with a couple of land snail families belonging to other groups, but for the most part, there's about 100 families here. The land snail family, as you can see from Hawaii, kind of spans this entire tree from the very top, from the Amastrids to all the way down from the Succineids. So we have relationships of other snails from all over the world. However, we call this this harmonic fauna is because we don't have all the representatives of these families from any of the closest continents, but we only have 13 families, right? But out of 13 families, a spectacular radiation of species. So from 13 families, about 21 colonization events, we have over 750 species, which is what we call a spectacular radiation, which is also patterns that you see on a lot of other isolated islands, and not including just snails, but plants and insects. So in Hawaii, you can find snails from the highest point of the trees to the lowest part of the grounds and the rock rubble. They also have a lot of different reproductive strategies. It's because they do span the land snail tree of life that, there are a lot of strategies that, they retained from their ancestors. We have some snails that are dioecious, where you need a male and female to have them reproduce. We have some species that are hermaphroditic. The majority of the species are hermaphroditic, which also gives them a advantage of colonizing Hawaii, because you don't need to have another opposite sex mate to actually reproduce. And then also there are different ways that they lay their eggs. So some are oviparous, they lay their eggs outside of their bodies. And some of them are ovoviviparous. It looks like they're having live birth, but that's not really true. Live birth is only relegated to like the mammals. But it looks like it's because the eggs are still inside the shell in the mommy's body. And when they hatch within the shell, and then the babies come out. So it looks like they're having live birth, but actually technically they're not. We also have a diversity of shell shapes in Hawaii, from the beehive ones all the way to the helical spiral, or what my kids call them, cinnamon buns, and the cone snail, which is like the ice cream cone shells. And this family, the Amastridae, as you can see, has everything from cone snails to the cinnamon bun shells and everything in between. A lot of our shells are micromollusks. So as you can see on the lower left, those are adult snails, and that white piece of thing is a rice grain. So you can see how tiny some of our land snails are. And the largest land snail we used to have in Hawaii was about 3 inches. Unfortunately, we haven't found any of these snails around, and likely they are extinct. And something very different that the Hawaiian land snails do differently from a lot of other snails is that our land snails do not eat live plants or live seedlings. They are basically the cleaner of our forests. So here is a 1 second old baby snail that hatched in our captive live collection here. And it's scraping off fungus off of the injured parts of the plants. So they're basically like our plant doctors. They keep it clean. They scrape off fungus and biofilm. They're also break down all the plant and animal waste matter. And like a lot of my kids say, the snails poop out fertilizer packets. And that's exactly what they do. And so we need these little critters to help keep our forests healthy, clean, as well as helping with the nutrient turnovers. Because we're in the middle of the Pacific, so we don't have a lot of resources and nutrients that gets into our system. So we need our recycling to be helping us out. When I first started, a lot of our snails were thought to be extinct, about 90%. Unfortunately, this was a misconception. And so one of the lessons that you want to learn is always look at the data. So most of the conservation in Hawaii kind of only targeted the pretty charismatic ones, but they only made up 13% of the fauna. About 90% of them are gone. So they expected, oh, if they're gone, about 90% are probably gone for the rest of the snail fauna too. And so that's a huge misconception, because if you don't think they're alive, you're not going to spend any resources or management to be saving any of these snails. So that's a knowledge gap. You can't conserve or protect what you don't know about. So again, what do you need to know to be conserving them? First, it's the systematics. What are they? Where are they found? What are their historical and current distributions? And then, until then you can figure out what habitat requirements are needed to be conserving them. So over the last 15 years or so, we have been doing a lot of surveys throughout the Hawaiian Islands. We employed several techniques to try to ID them, because a lot of them have not been studied for 50 to 100 years, or since they were first described in the early 1800s. And we use morphology, genetics, as well as internal anatomy to help us to be identifying them. And of course, the amazing museum collections all over the world, particularly the ones at the Bishop Museum. So we have some good news. Not 90% are extinct. We were able to rediscover about 200 species that are still threatened, highly threatened, but still has a fighting chance for us to still study and try to preserve them. So we have about 300 out of 750 left. So still not a lot, but still a lot more than we first started. And here are some of the amazing snails that we have pictures of now. So these are like one of the first digital images of some of these of snails. A lot of them were hand drawings, right? And a lot of these really great references, but you lose a lot of detail and information from just drawing. So that's why museum collections, as well as digital photography, is very important to capture what they look like, so people know what they are trying to identify when they go back out into the field. And then we were able to describe some new species. So about 10% of the things that we discovered were new to science. And people are like thinking, oh, great, you can, describe new species. Yes, it's fun to describe and put names to it, but it's also a very important practical aspect. If the snails does not have a name, you cannot provide any management plan for it. So I can't say this is Auriculella species 32. That is not going to help with anything, because you need to have an actual description. It has to be a valid name, and it has to be officially recognized before we can put any management resources or conservation management or research towards these. So out of all of the new species, we picked these two. It's because the top one is endemic to the Waianae Mountains on Oahu, and there's only one wild population left. It takes, they lay only one egg at a time, and it takes nearly 2 months for it to hatch, which is why they're going extinct. It's not the best strategy for them to survive at all. So, and then this bottom one, Endodonta, that's the last known Endodonta species on Earth. So if this one goes extinct, the whole genus goes extinct. It is only found in one little isolated atoll north of the main Hawaiian Islands. So if a tsunami, a hurricane, or invasive species get onto this island, they can easily wipe out this last snail of Endodonta. And so we are at least putting flags on these two species and telling conservation management as well as the government, please pay attention to these two snails because they can likely go extinct in our lifetime or within the next decade, unfortunately. So here is an overview of the extinctions of each of the families, which as you can see, most of them is more than 50%. And most of the ones that have gone extinct are the ones primarily found on the ground because due to habitat destruction as well as invasive species, they go first. And a lot of the snails that we do have left are in refugias in the highest parts of the mountains on the highest part of the trees. And of the 300 species that we have left, we are expecting 100 to go extinct within the next decade unless we do some drastic conservation management efforts to be helping with these snails. One of the things that we are trying to do is put them in lifeboats, which is basically little boxes in our museum. So we have all 23 species that we are rearing in the collection, and some of them are extinct in the wild. And if you want to learn more about the program or if you want to donate to the Hawaiian Land Snails Conservation Fund, here's a QR code that you can snap a picture of. And all of the donation goes towards buying paper towels and supplies for our captive rearing because it is entirely volunteer based. We're lucky to get some funds from the state, but for the most part, it is still primarily run by volunteers and a research affiliate who doesn't get paid at all to be caring for these snails. It also goes towards research as well as internship opportunities for the students. I want to show you a very quick video of some of the snails we have in captive rearing. So this is a Neucombia canaliculata, endemic to the island of Maui, with only one wild population left that we know of. This is Lamonella sanguinea, endemic to Oahu, extinct in the wild. This is Acatinella sarbiana, also extinct in the wild. Auricula lamelliata, endemic to Oahu. This is our undescribed Succinia species. Mastra micans, also extinct in the wild. And Kaala subretia that we rediscovered in 2008, and the last known record was in 1960. And as you can see, these beautiful speckles on the shell, it comes from the body. The shell is translucent. So once this species goes extinct, you will never ever see the beautiful red on the head or the beautiful gold speckles that you see through this translucent shell because it will no longer be alive. So with extinction, we lose a lot of information, we lose a lot of beauty, and we lose a lot of the environmental services that these little critters provide for us to keep us happy and healthy. So in addition to doing a lot of native species work, we also, as you know, as I said earlier, we're the extinction capital of the world. It's because we have a lot of invasive species that have been brought to Hawaii either on purpose or not. And so there are many ways that you can get to Hawaii, right? You can have intentional introductions. People can bring in snails or plants or other animals for food, for religious, cultural reasons, as pets, because, you know, some of them like to rear snails in terrariums, as well as for biological control, right? But there's also a lot of ways to get into Hawaii through unintentional introductions. So a lot of the plants that are brought in, the Christmas trees, when you are moving from, you know, from one area to another, we had a really bad snail hitchhike on a barbecue grill from Florida to Hawaii. Luckily, it was only one snail and it was not pregnant and we killed it. But we don't need it here. And also this picture right here, this was a car that was brought from Europe to be sold in Hawaii. And you can see Otala lactea, this snail, this really, really bad milk snail. in these undercarriage of these cars. So yes. And this is a simulation of 24-hour global shipping tracking. You can see just for this shipping, how much stuff we're moving around across the world. This is an older paper from 2009, but you can already see the trajectory of the amount of shipping cargo that we have increased from 1975 to now, right? We've increased our bulk cargo, our oil, our freight, and as you can see on the right graph, we're also transporting a lot of plants, invertebrates, and mammals around too. You can even see this same graph pattern for all of the non-native land snail species we are tracking in Hawaii. You can see this exponential growth as we increase our cargo and shipment everywhere. And people are like, oh, you live in Hawaii, don't worry, you're not gonna get a lot of things established here. It's because it's hot and tropical. But people don't realize that we actually have snow on the big island up in Mauna Kea. We have coastal sand dunes on several of our islands. We pretty much have every single ecosystem in the world except for Arctic tundra. So if you come from Europe or Africa or any of these places, you will find a nice habitat to establish in Hawaii. And the ones that are super, super, super bad are the ones that are coming from temperate regions. So, in Hawaii, we don't get really nice pine trees. So we ship a lot of these Christmas trees from the Pacific Northwest. And of course, with the Pacific Northwest, there's a lot of slugs that we don't have in Hawaii. All of our snails in Hawaii have shells on them. So if you're a shellless snail, you're invasive or non-native. We also did a lot of survey work in horticultural areas, and we found that one of the worst snails that are coming are from temperate regions. Is because these snails can withstand really, really, really hot weather and really, 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 really cold winters. And so they have a much higher range that they can establish in Hawaii and wreak havoc. Unlike our native snails, though, they do eat plants, live plants, and live seedlings. So they have pretty much ate a lot of our endemic native plants, and extirpated them in many areas. So we don't need more of them. We don't need any of them. And we also have invasive snails that are carnivorous or molluscivorous. And so one of the snails that were brought to Hawaii was Euglandina rosea. It was used to control the giant African snail. But as you can see in the upper right-hand picture, that's the size of a giant African snail and the size of Euglandina. So instead of controlling the giant African snail, which we still have a lot in our islands, it started to eat all of our native snails, which are bite-sized morsel, perfectly in line for their mouths. So this is a Euglandina rosea. We did not feed it a native snail. This is another invasive snail from Asia, the Bradybina similaris. But that is what it does. And that is how it extirpated a lot of our snails. And talking about wanting to have information for knowledge graphs for systematics, they thought they brought one species of Euglandina. And we found out, based on our morphology and genetic work, working with John Subsiski at the Florida Museum, there are 3 species that have been called Euglandina rosea that have been introduced to Hawaii. And when we tried to assess rosea in Florida, we found 11 cryptic species. So knowing your species and what they do is really important, because we also found out one of the species has a larger range in Hawaii and also reproduce much faster. So once we are now able to distinguish the different species, we are now able to map the reproductive anatomy and their life history, and we know one is a much larger threat than the other. So knowing your taxonomy and your systematics is very, very crucial if you want to be saving things, but also figuring out how to prioritize to eradicate species. And one of the things that we try to do to help the community as well as other researchers, because a lot of these snails, like I said, are going extinct faster than any of us could ever study them before they go extinct. And so we were able to get funds from NSF. This is a Pacific Island Land Snail TCN. And I believe you folks are also part of a couple of TCNs to help digitize your collection to be put into a larger collective. I was a little bit selfish when I put this grant in, and it encompasses 5 other natural history museums, which are absolutely amazing. The Field Museum, ANSP, Michigan, Florida Museum, and then us. I mean, am I missing one? Sorry, I usually have these logos. I actually missed the logos in here. I'm really embarrassed, and my collaborators are probably going to be teasing me endlessly for this. But
I think you named 5. I think you named 5.
Okay, and the Bishop Museum. Okay, whew, it's like, where are their logos? I'm very sorry. But they're also very good friends of mine, so I hope they'll forgive me. But yeah, so out of our 6 collective museums, we have records for 3.6 million specimens, 261,000, like more than 250,000 lots, encompassing these species. And to date, we have all of these up in this Pillsbury portal. I wanted a one-stop shop, honestly, for my research to be helping these snails to have their historical distribution, to have all of the field notebooks, understanding all of their habitat requirements.
Nori, I'm sorry to interrupt.
Yeah.
Can I ask, but just for folks on the who may not know, what does TCN refer to?
Oh, that is the Thematic Collections Network. So it's basically a network that the NSF is supporting as many collections as possible, trying to push information out to help with particular research questions or problems. So in this case was trying to put out as much information for that is needed to be conserving Pacific Island land snails. So everything from historical distributions to their systematics to identification. We try to put all type materials up, all of their images, so that anyone wanting to be studying them has all the materials that we possibly have on hand for them to be studying their evolution or their ecology, pretty much anything that they want on any Pacific Island land snails.
Gotcha. Thank you.
Yeah. And so here's a little tour of our sites. So you can search by collections, by habitat. So a lot of things that are already associated with a lot of our specimens. We can type in a species name. So we put in Acatenella bulamoides. You can have different ways to display it. And so it also includes all of the chrysonomies, the subspecies, all of the old combination of names and also the current established name or valid name. So this one from the Bishop Museum, we have a little over 1,000 records. We are trying to geo-reference them based on old maps, collection events, descriptions, so that it can help us start to understand, whether populations have expanded, where we can translocate some of our populations to expand the species ranges. So this is what I really, really love about this, is basically using all of our collections to build me a treasure map. And then if you click on one of the dots, it has all of the specimen information, the type image, if we do have them, all of the associated labels. We have also scanned the ledgers for all of our museums. So they're also linked to each of these records. And so like this ledger says that it's from this particular area, from Welch, that was collected by Emerson. And yes, there's a map, there's a field notebook that is attached to it. And yes, we have scanned it. So then we have this field notebook that we scanned, and it tells exactly where they have located it. So now X marks the spot. Maybe we can go there, maybe it's still alive. Or maybe we can translocate some species back there. So as we collect more and more information, as we build up and incorporate all of these information, we hope that we can really truly understand more of the species so that we can help conserve them. So over the last 10, 15 years, we've been trying to tackle these first 4 shortfalls. And now we currently are going into these other 4 to expand and fill in some of these knowledge gaps. So we are now working with the state to, did it change to the next slide? Not quite yet. Next slide. There you go. We have a parasite screening. So even though we're working with snails, right? Snails live in the world. So we're trying to understand their nematodes, the bacteria, the fungi, everything that is associated with diseases, so that we can help them possibly better survive in the wild. But also we have sometimes disease outbreaks in the lab, so that you can see them isolated in these little plastic Petri cups. So the more we understand things that are growing on them, with them, things that we're bringing in from the community, the better we have a better chance to try to be conserving and increasing the survivorship of some of these species. We just got this grant. It's a $1.6 million grant that we just got from NSF, and this is our first summer that we started. It started on May 1st. We are now going to the different islands, going to the different forests, having 8 plots in these little areas, and counting every single snail and every single plant in these 5 by 5 meter plots, so that we can start understanding plant preference, so that we know areas that we need to restore certain plants, so that we can translocate some of these snail species. Also, we don't know what a lot of these snails eat. So we unfortunately have to take wild plant cuttings from the forest to be feeding them in captive rear, right? Because we don't know what fungus or biofilm they're eating. And so we're also swabbing all of these plants, so that we are getting a sense of what they need to be eating, so that hopefully we can make some snail chow for them to be eating in captive rearing, but also seeing what they need in the forest, because there are areas that we reintroduce them. They do very well in captive rearing. They make lots of babies. But once we put them back out on the forest, they don't. So it's like a slow extinction. And we need to know, is it something about the forest? Is it something that they're lacking in the food source, or is it weather? Because we also build predator-proof enclosures around them. So without any impacts of predators, they're still not making a lot of babies. Why? So we're hoping that this grant will provide us some insights. And then we're also partnered with the state. So all the resources and research that we provide, they're going to be implementing them into their conservation plans also. So this is a great synergy of bridging actual research and putting them to actual applied conservation management. And last but not least, the Australian. There's no point in doing any of these research or publications if we do not interact with the community, because the knowledge will basically die with us, and that is useless. It is useless and a waste of our resources, our money, and of our time if we do not incorporate the community and teach the next generation. So in all of our grants, we have put in funds for having interns. We try to do as much educational outreach. We do taxonomic workshops. We train students to do science communication, which is very hard, right? To actually communicate your science to a whole different types of different communities, age groups, knowledge groups. But if you can, I tell the students, if you can explain to your grandma why it's important to be saving snails, you got the skills to be communicating the science necessary for them to care. And it took a while because, snails sometimes get a bad rap, right? They're gross, they're slimy, they eat up all of our vegetables, but we were able to at least provide a lot of the knowledge of the importance of snails, of why we need to keep them in our society, and not just for our planet, but for our society and culture, right? And we were happy to announce that the governor actually named this year in Hawaii Year of the Kahuli. Kahuli is one of the Hawaiian names for land snails. So we have festivals, and also one of our high schoolers back in the days wanted to raise awareness and appreciation for these snails. And so he drafted a resolution to submit to the legislator to also vote for a state snail. So we're happy to have that. And then also a lot of kids like to play Pokemon. And so one of my other interns said, why don't we make snail profiles and make trading card games? And so all of these are the efforts is because we've actually given the tools for these students and these interns, and they're so passionate about these snails. They love these snails, and they have like the best ideas on how to raise awareness and connect with the community. So including having resources for our research and our museums and our universities, we also need to remember to put the resources into the community engagement, because it really, really matters. And so I kind of want to end this last part. This is one of my kids. We put them in all of the snail museum statues and structures that we find in any museums that we kind of visit. But it's true. Unless someone like you cares an awful lot, nothing's going to get better, and it's not. And we need to start small, right? Because in the end, we will only conserve what we love. We will only love what we understand. We will only understand only what we are taught. So with that, we have a lot of people that we would like to thank, because it is not only us, it is a community. And that's what we try to teach the students. Science is not done in a vacuum. You need to try to stretch your arms and get involved with as many people, any entities as you wish, because even if you save the snails, it doesn't matter if you don't save the forests, if you don't save our waterways, and the mountains are connected to the sea and vice versa. So if you don't take care of one habitat, it's going to connect to another. So it doesn't matter if you are going to end up working on snails, just be aware of where you live and how you interact with the community, both people as well as the forest. And with that, I know I kind of sped through that, but if you have any questions, sorry, is that not too fast? I'm sorry.
That's great. That's great. No, thank you. Thank you. Good advice and a great presentation. And you're obviously working on a lot of amazing things. We have I know there's one question in the chat. I had a question actually. So, your scope of work is very broad, and kudos, by the way, for getting the declaration for the year of the land snail. That is like completely amazing and something that we need to aspire to here. But I know just from emailing with you several times over, recently, that you're in the field quite a bit.
Yes.
Could you talk a little bit about, the kinds of things you're doing when you're going out into the field?
Yeah, so see, sorry, I have multiple monitors, so it looks like I'm not looking at you because I'm looking at, so I'm trying to shift it to my front monitor so I can look like I'm looking at you. Yeah, so when we're on the field, it's pretty much continuing our monitoring efforts. As you can see from the map, there are a lot of areas we still haven't went to, right? And we also want to continue monitoring because unfortunately, there are some areas that where we're so excited that we were able to refine or rediscover a population, and we go back there and there was a flood and there's a landslide now, and that plant that all the snails were on were white. So it's a continuing thing that we need to be doing, and it's tough. I wish I could clone myself. I need me as a field monkey, I need me as a lab rat, I need me as the dancing bear to be talking to people, and then I need to go home to my boys.
Right.
So it's not easy, and then also your curators and Jose, they do all of it too, right? And that's a great thing about being in a museum. You have so many opportunities to do so much different types of work, but it can also get a little bit overwhelming when there's not a lot of support and help, right? But that's the reason why you need to engage the community, because without these volunteers, we wouldn't be able to do what we're doing.
Right. Yeah, we've got a very, very fledgling effort here to working with community members to just document land snail species that are in this area, which very little of that has been done. So just using our naturalists and going out on biodiversity walks and doing that kind of documentation. So
yeah, that's a very good start. But and it's also a very fine line on how to do this well. We also need to continue to collect and voucher in museums, right? And people are like, why are you collecting? They're endangered. But the thing is that we need to collect is because the things we have in the museum is basically a particular chapter in the life of these species, right? When you have these photographs, you cannot do DNA on them. You cannot understand if there are any pollutants that were in the environment that are now established in their body. You cannot see if there were any diseases or when diseases were established. So that's why these museum collections are so important, is because it's a snapshot in this timeline of these species. And if we stop, right, we don't know in 50 or 100 years whether we needed to understand their genetic diversity or understanding that maybe there is something that can cure some disease now, right? So there is a responsibility on how we collect, how we voucher, and the information that we need to do to preserve these collections in perpetuity, right? Because these are not my collection. These are the community's collections. And so we need to be very cognizant that photographs and having a community is the very first step. But we as a museum also have a responsibility for society to be doing the best up-to-date work that we can to monitor and catalog this, tree of life we have on Earth, right?
Very well said. Very well said. And along those lines, at the Bishop Museum, what is the collection of say terrestrial mollusks? About how many specimens, about how many examples are in the collection?
So we have nearly about 3 million specimens and about 3 million uncatalogued. So if any of you, I don't want to steal from your pot, Sam, but if any of you guys want to come to Hawaii and help me catalog all my back catalogs,
I wouldn't want to do that. Yeah. Wow.
So we can't pay you back, but I'll feed you.
Yeah. So that's a major collection for folks who don't, as far as organizations that have collections like these, that's major. So that's terrific. Great resource. All right. A question or two in chat.
Oh, for the first one is what is your hand signal in the group photos?
Yeah, Lori's asking what yeah.
So that's pretty much our sign for like peace. Hang loose, it's all good. Thank you. So this is like, what's up? So this is basically our local shaka sign.
Yeah. Nice.
It'll go into so many ways, like thumbs up, it's okay. Is it okay? Yeah, it's okay. How's it going? Hi. That's basically our sign here in Hawaii.
Very nice. All right, so you see John's question there at the bottom. Maybe you could read the question and then answer it. Yeah.
Yes, John, we would love to find Carelia.
Or first read the question. Read the question first for folks.
Is research being conducted in the field on Kauai to hopefully find living Carelia snails? Yes, we are holding out hope that there is a Carelia out there, but very unfortunately, likely not. There were ground snails, the larger snails. So the rats, the pigs, the mongoose, Euglandina, habitat destruction, all of them would have wiped them out. We are hoping that there is some isolated valley in Hanalei on Kauai that there's a population left. We're truly, truly hoping, because we have rediscovered snails that haven't been seen since like the 40s and 60s. And so I usually tell people, and even our conservation manager collaborators, if you find a live Carelia on Kauai, I will treat you to any dinner, any place in the islands, anywhere you want. Because it's gold.
That's your commitment to science.
Yeah, but unfortunately it's likely gone. But we have been able to get genetics from some of the bodies of Carelia from our collection. So that's one thing that's great about this Bishop Museum collection, is that these historical collections never was preserved in formaldehyde, which is great for morphology, but that molecule binds to DNA, so it's very difficult to uncouple these two. Because we live in the middle of the Pacific during that time, we couldn't really get formaldehyde here. So the curators had their interns go into the pineapple fields, steal pineapple and make moonshine. So they pretty much ended up in ethanol. And so that's a great, treasure of these specimens we have in these alcohol collection, is never touched formaldehyde. So we're able to at least uncover some of the things that we have lost, So.
Yeah. Nina asks, how do you integrate citizen science? It's important and valuable, but the data is limited. I guess we were sort of talking about that a little bit before. So that's the question. Yeah.
Yeah, so it's definitely hard. So that's why, we wanted to have that portal for all of those historical information, as well as have all of those pictures. So it's like iNaturalist, but it has a lot of data integrated with it, as well as researchers who actually, kind of like went through all the collection. To integrate citizen science, first thing is to get them into the collection, right? Get these volunteers in, know what information you need to be collecting. They become our eyes of the forest, our boots on the ground, right? The more people we have out there knowing what we do, the information we need, then the more information that they can help us collect also. Doing workshops, doing festivals, it is very time consuming and very energy draining, considering I'm actually an introvert, I actually like my alone time a lot. But it's necessary. People are like, well, it's hard. And I'm like, well, extracting DNA out of a 100-year-old specimen is hard, but it's necessary. And engaging with the community, citizen scientists, is just as necessary. So that's just my priority level. It's not below or ever an afterthought. It's part of our program here. So
Do you have a pretty good group of volunteers now working with the collection?
yeah, no, we do. We have I even have high schoolers that start off sophomore year and stay for 3 years through their senior years. Like, you can go surfing, you can go sleep in, but they're actually coming in here because they actually care about their snails.
Amazing.
They actually call them their snail overlords, right? So it's very, very quick that they start taking over your life. But I call them my little minions. They call themselves baby snailers. So yeah, no, we have high schoolers all the way up to people who have retired. And we take anybody, right? And I found out retired accountants are the best in digitizing. They're great with numbers. They're so detail oriented. They're fantastic. If you can somehow get some accounting firm and tell them we want your retirees, take them. They are so good. And so we tell them, yes. Yes.
Are the high schoolers coming to you as individuals or as part of a sort of a partnership you have with any schools or anything like that? Or they're just interested in getting connected with the museum on their own?
Yeah, so it's a little bit of a bunch of them. Some is like, their mom is making them to do volunteer work, and then they want to bring them friends, and then they get hooked, which is fantastic. Other times, like I said, we have our external grants where we have internship programs, and then we are able to advertise for them. But a lot of it has been great. I guess we're not too bad of a place to work, so a lot of it has been word of mouth. So I've hardly had to advertise, and I'm consistently getting a lot of volunteers, but we also do a lot of outreach, right? We have festivals. So any opportunity that we're able to engage with the community, I have a QR code for our volunteer sites for them to actually sign up. And we are actually able to sometimes we actually have to turn some away because we just don't have enough bench space. Or they want to come to the weekends, and, I have to kind of do a work-life balance. I don't want to bring my kids to work. So I'm like, no weekends. I'm not working on weekends.
Right.
So.
Right. Jose has a question. Do you continue efforts to eradicate rosy wolf snail species from the islands?
It's so tough. It's actually tough to eradicate any snails in Hawaii, right? Because remember that Sanamatofan tree of life I showed you? Our snails are pretty much spanning it. There is no bait that won't kill our native snails. There's no gene drive or genetic way of killing these invasive snails. So it's very difficult. It's a lot of times just trying to build these what we call mollusk mansions or snail jails, however you want to call it. But these are predator-proof fences in these little pockets on our forest that excludes these predators, but that's in no way for them to be living, but that's probably the way that we can keep the rosy wolf snails from eating some of our snails. A lot of it's manual control. We really don't want to put snail bait out because, like I said, it's going to be killing a lot of our other snails too. We can't get rid of giant African snails. I know you guys are still trying to eradicate your population. Get rid of it as soon as possible as you can, or else it's just going to be
Yeah.
heavy
It's a pro
damages.
it's a problem that just keeps popping up, it seems.
Yeah, which is why
Whack-a-mole.
they're on the list of the 100 worst invasive species in the world, right? Euglendina
Mhm.
rosea, as well as the giant African snail are on that top 100 list.
Mm.
Yeah.
Yeah. Okay. Well, Norine, thank you so much for your work, and thank you very much for being with us for this presentation. And for an introvert, you're a fantastic presenter.
Thank
So
you.
thank you. It was interesting and informative. So thank you very, very much, and keep up the good work.
Thank you.
And
Thank you for inviting
yeah,
me again. I really appreciate it.
yeah, and we look forward to having you come visit again, when we're back up and running, and we'd love to get symposium going again, like
Love that.
last time you were here. And so thank you. Thank you everybody for joining the program. And this is being recorded and will be available within a couple days on our website. So if it's something you'd like to share with others, you can go to our website in the education section and find it and share the link. We'll also send it out by email within a week or two as well. So thank you, Nori. Have a good
Thank you so
rest of your
much.
day.
Thank
Yeah.
you.
Okay. Everybody,
Lunchtime.
thanks so much. Okay. All right. Bye.